Welcome back to our online classes. We have started with second chapter, that is sexual reproduction in flowering plants, in which we have completed up to male gametophyte, that is up to pollen grain. We have studied the structure of pollen grain and we have seen that it can be preserved, it can be stored for a long period of time. Okay, and today we will be starting with the new topic. Today we will start with the new topic, that is development of female gametophyte. So let us continue. We are starting with development of female gametophyte. So shall we start, children? Okay. Now, female gametophyte development requires the study of pistil, then Megasporangium, megasporogenesis. Okay, so let us start with the study of pistil first. Pistil, as you have learned, it is also called gynosium, and it is the innermost hole. It is the female reproductive part of the flower. It is also called carpel. Okay. So the gynosium represents the female reproductive part of the flower, and it may consist of one pistil or carpel, which is known as monocarpellary. If only one carpel or one pistil is present, it is called monocarpellary. If two are there, if two pistils or two carpels are there, then it is known as bicarpellary. Three carpels, then it is called tricarpellary. And if there are many carpels, then it is called multicarpellary. Okay. Now, pistils may be fused. If it is fused, then it is known as syncarpous. Okay, if many pistils are there, right, and if the pistils are fused, then it is called syncarpous. And if the pistils are not fused together, that means they remain free, then it is known as apocarpous. Okay. Now, syncarpous is observed in papaver. Example of a syncarpous, that means fused pistils or carpels, is papaver. And free is found in rose. It is also found in Michaelia. Okay. Now, a typical pistil consists of a hollow basal swollen part which is known as ovary. It is hollow, and a basal swollen part which is called ovary, an elongated style and a terminal stigma. Okay. And the stigma serves as a landing platform for pollen grains. Inside the ovary is the ovarian cavity. Okay, inside ovary the cavity is present, which is known as ovarian cavity, and ovarian cavity is also known as locule. We have already studied this in 10th class, in which placenta is present. Megasporangia or ovules arise from the placenta. About placentation we have studied in 11th standard, and number of ovules in an ovary may be 1 as in wheat or paddy and mango, to many as in papaya, watermelon, orchids. Again, this is the revision. We have already completed this in 11th standard. We have studied this in 11th standard. Okay, so let us move to the next slide. Okay. The next slide is about the Megasporangium. Now, Megasporangium is also known as ovule, right? So, we'll be studying about ovule in this chapter. Sorry, in this slide. The ovule is an integumented megasporangium within which the meiosis and megaspore formation takes place. It is attached to the placenta by the means of a stalk which is called funicle. It develops into a seed after fertilization. The junction between an ovule and funicle is called hilum. This one, hilum. Right. So what is it? It is the junction between the ovule and funicle. And funicle is the stalk by the means of which the ovule is attached to the placenta. Okay. Now this hilum becomes a scar later on the seed. Each ovule has 1 or 2 protective envelopes which are called integuments. Each ovule has 1 to 2 protective envelopes which are called integuments. The inner layer protective envelope is known as inner integument, and the outer protective envelope is known as outer integument, which encircle the ovule except at the tip, where a small opening called micropyle is present. So in the micropyle region only the integument is not present, otherwise the integuments covers the complete ovule except the micropyle. The basal part of an ovule just opposite to the micropyle is known as the chalaza. Enclosed within the integuments is a mass of cells which is known as the nucellus. Cells of the nucellus are rich in reserve food material. An ovule generally has a single embryo. An ovule generally has a single embryo sac or female gametophyte develops from a megaspore through reductional division and located within the nucellus. This development will be studying in the later slides. Okay. This is about the diagrammatic view of a typical anatropous ovule, wherein you can find that there are integuments, right? These are 2 integuments. This one and this is the another. This is outer integument, this is inner integument. The innermost layer of the cell is known as the nucellus. Okay. Then this is what? This is the embryo sac. It is already labeled, you can see. So this inner part, this is embryo sac. Okay, and inside embryo sac there are 3 antipodal cells. Okay, these are antipodal cells, and you can see the integument covers the complete ovule except the region which is called the micropyle. In the micropyle region there is no integument. Okay, then yes, this is what funicle, which connects the ovule to the placenta, right? So this is funicle, and just to the funicle. Just beside the funicle there is hilum. Okay, so this end of the ovule is known as chalaza, which is opposite to the micropyle, and here micropyle an opening is present. It is known as micropylar end or pole. Okay, this is also known as chalazal end or chalazal pole. You can say anything. Okay, now we were talking about the embryo sac. Inside embryo sac there are antipodal cells. This is the secondary nucleus. Then there are synergids. These two cells, this one and this two, that are called synergids, and in the center there is the egg cell. Okay, and this complete structure is what? It is embryo sac. Clear? Okay, so let us move to the next slide. Fine. Now, depending upon the shape and orientation, the ovules of angiosperms are classified into different types. The first one is the orthotropous or atropous, where the micropyle, chalaza and funicle are in a straight line. Okay, example Piper, Polygonum, Cycas. Clear? The second one is the anatropous, where the body of the ovule is completely inverted, and 82% mostly it is found in 82% of the angiosperms. According to your textbook, it is 90% of the angiosperms family have this anatropous ovule. Then the next one is the Camylotropous, where ovule is circled more or less at right angle to funicle. Example is Leguminaceae cruciferae family. Next one, Hemianatropous. Okay, Hemianatropous, where ovule turns at the angle of 90 degree upon the funicle. Here diagrams are also given. See, you can see here it is anatropous, where it is orthotropous, where the micropyle, chalaza and funicle are lying in the straight line. Micropyle, chalaza and funicle, they are lying in the straight line. Okay, the second one is the anatropous, where the body of the ovule is completely inverted. You can see inverted ovule है यहाँ पे, जहाँ पर micropyle नीचे आ जाता है, chalaza end ऊपर है. पहले वाले में क्या है? In the first case, this is micropyle and this is chalazal end. पर ये पूरा inverted है, तो inverted होगा उसको क्या कहेंगे? Anatropous. फिर थर्ड वन है Camylotropous, जहाँ पर ovule is circled more or less at the right angle to the funicle. ठीक है, तो ये funicle है और इसके right angle पे यहाँ पर पूरा ovule present है. So this is Camylotropous. Okay, the fourth one is Hemianatropous, where you will find that ovule turns at the angle of 90 degree upon the funicle. 90 degree पे पूरा ये turn हो रहा है. ठीक है, that is Hemianatropous. The fifth one is Amphitropous, where the embryo sac becomes curved like horseshoe. ठीक है, horseshoe जैसा कर्व हो जाता है. You can see here horseshoe जैसा ये कर्व दिख रहा है आपको. हाँ, embryo sac कर्व हो जाता है. So it is what? Amphitropous. An example is lemna, poppy, elsima, etc. And the last one is Cercinotropous, where the funicle completely surrounds the body of the ovule. यहाँ पर देखिए ये funicle है तो वो पूरा ovule को कवर करता है, पूरे ovule के बॉडी को कवर करेगा. So that is what Cercinotropous. I hope you have understood this different types of the ovules based on their shape and orientation. Now we will start with the topic megasporogenesis. The process of formation of megaspores from megaspore mother cell or MMC, मेगास्पोर मदर सेल या फिर MMC में से मेगास्पोर बनाना, उस प्रोसेस को कहेंगे मेगास्पोरोजेनेसिस. ओके, इट अकर्स इनसाइड द न्यूसेलस. ठीक है, न्यूसेलस के अंदर होता है of developing ovule of angiosperms. The process begins very early when nucellus is not completely surrounded by the integuments. The MMC or megaspore mother cell, a large cell containing the dense cytoplasm and a prominent nucleus, enlarges in size and divides by meiosis to form a tetrad or 4 haploid megaspores. ठीक है, तो यहाँ पर क्या होता है, मेगास्पोर मदर सेल और MMC विच इज डिप्लॉइड, सॉरी हियर द स्पेलिंग मिस्टेक, DIPLOID, डिप्लॉइड. ठीक है, वो कहाँ पर होता है, न्यूसेलस के सेल्स के बीच में होता है. और ये कैसा सेल है, लार्ज सेल है जिसके पास डेंस साइटोप्लाज्म है और अच्छे से दिखने वाला न्यूक्लियस है. तो वो क्या होगा, वो एनलार्ज होगा साइज में और फिर वो मेयोसिस सेल डिवीजन करेगा और उसकी वजह से क्या होगा, देयर विल बी फॉर्मेशन ऑफ 4 हैप्लॉइड मेगास्पोर्स. ओके, now let us study about the female gametophyte, and in general the development of the female gametophyte, which is also known as the embryo sac, is monosporic as in Polygonium, and in this type of development only one megaspore which is situated towards the chalazal end remains functional, while the remaining 3 megaspores gradually degenerate and finally disappear. ठीक है, तो जो मियोसिस से 4 टेट्राड मेगास्पोर बने हैं, 4 haploid megaspores which are formed, out of that the 3 will degenerate and only 1 will remain functional. The 3 which are near the micropyle will get degenerated and only the 1 which is near the chalazal end will be functional. Okay, and the degenerated one will then disappear. Okay, now let us see different stages of development of female gametophyte. The functional haploid megaspore is the first cell of female gametophyte of angiosperm, and it enlarges in size to form the female gametophyte, which is also called the embryo sac. Its nucleus undergoes mitotic division to form 2 nuclei that move to opposite poles, forming 2 nucleate embryo sac. The 2 nucleate embryo sac undergoes 2 more sequential mitotic division, giving rise to the 4 nucleate stage and later the 8 nucleate stage of embryo sac. This stage comprises of a micropylar end and a chalazal end with 4 nuclei at each end. 6 of the 8 nuclei are surrounded by the cell walls and gets organized into cells. 3 cells present towards the micropyle end grouped together constitute the egg apparatus, that is 2 synergids and 1 egg cell. 3 cells of the chalazal end are called the antipodals, and the large central cell is formed by the fusion of 2 polar nuclei. Thus, a typical angiospermic embryo sac or female gametophyte at maturity consists of 8 nuclei and 7 cells. I'll show the diagram now. See, this is the development. This is the megaspore mother cell which is present in the nucellus. Okay, and it is large, it has dense cytoplasm and it has a prominent nucleus. Now this megaspore mother cell undergoes the meiosis, which results in formation of, this is completion of meiosis 1. At the end of the meiosis 1, there will be formation of 2 cells, right? So it is known as megaspore dyad. Then further, this diagram shows meiosis 2, which is nothing but mitosis, right? So it results in formation of 4 megaspores or megaspore tetra. This is the micropyle end and this is the chalazal end. So out of 4, the 3 which are near the micropyle end will get degenerated and only the 1 which is towards the chalazal end will remain functional. And further it will undergo division. It undergoes mitotic division. Okay, repeated mitotic division takes place without the cell wall formation. So one nucleus divides into 2. Okay, further it divides and produces 4 nuclei. This 4 nuclei undergoes mitotic division and produces 8 nuclei. Now all these 8 nuclei move towards the poles, that is towards the micropylar end and towards the chalazal end, and one by one they come to the center. Okay. Now further they will move towards the poles, okay, and only one from each pole will remain in the center, which are known as the polar nuclei. ठीक है, तो जो 8 है उसमें से 3 यहाँ पर रहेंगे. ये कौन सा एंड है? माइक्रोपाइल एंड है, और ये है चलाजल एंड या फिर चलाजल. तो यहाँ पर भी 3 सेल्स रहेंगे, यहाँ पर भी 3 सॉरी नॉट द सेल्स बट द न्यूक्लियाई. 3 nuclei will remain at the micropyle end and 3 nuclei will remain at the chalazal end, whereas 1 nucleus from each pole will travel and will settle in the center, which are known as 2 polar nuclei. I hope this is clear. After this, there will be division of the cytoplasm, which will result in formation of 3 antipodal cells towards the chalazal end and 2 synergids and 1 egg cell towards the micropylar end. ठीक है, और जो सेंटर में सेल होगा which has the 2 polar nuclei is known as the central cell. Clear? So this is the complete diagram of embryo sac, where at the chalazal end you will find 3 antipodal cells. Okay, these are 2 polar nuclei. और ये जो सेंटर का सेल है उसको क्या कहेंगे? सेंट्रल सेल. एट द माइक्रोपाइलर एंड the 2 cells are called the synergids and 1 cell is known as egg cell. Okay. I hope this is clear to everyone. In our next video, we'll be studying about pollination in detail. Children, if you have any doubt, you can ask me later. You can write the comments and you can ask me. Thank you.